Hello everyone. Today we are going to talk about a paper which recently came in Science Immunology, and we are presenting this paper as a Immunology Journal Club presentation here, and I will try to explain whatever I got from this paper, what I understood from this paper. If anything which is more you want to add, you can comment on this. And if any question if you have regarding this paper, you can ask in the comments. If it is possible for me, I will try to answer that question. I'm also still learning immunometabolism. That's why I'm choosing those papers to present in this online immunology channel club presentation. So today the paper which was recently published in Science Immunology and entitled is Mitochondrial-ER contact mediated by MFN2 and SERCA2 SCERCA2 interaction supports CD8 T cell metabolic fitness and function in tumors. So if we see this title, we can see here some terms like mitochondria and ER contact. Two molecules especially investigated in this paper is MFN2 and SERCA2 and their interaction. And this paper also talk about CD8 T cells metabolic fitness. And overall, these experiments were done in different tumors, especially mice, xenografts, and also samples from human tumors. So first, what we are about to do is we will discuss these terminologies and try to understand what this terminology means. So for example, if we see this terminology mitochondria-ER contact. So in cell, it has been known that mitochondria and ER both interact with each other, and in recent past, like last 2 decades, 2 or 3 decades, people investigated these interactions and try to attribute the functions to these interaction between the mitochondria and ER. So in this case, you can see, 1 minute, I will get this pointer and it will be easier for us then. Let me get pointer. Yeah, so you can see interaction between mitochondria and ER has been attributed to different function in the cells, like calcium transfer. Here you can see the calcium is transferred from ER, this is ER, to the mitochondria, and several proteins has been reported to involved in this tethering of mitochondria to ER and the transport. They facilitate the transport of calcium from ER to mitochondria. Similarly, the protein which you can see here, SERCA1, these transporters especially involved in the transport of calcium from cytoplasm to the ER, which is a non-function for those transporters. Apart from that, people also investigated different functions like the role of this interaction in lipid metabolism, in autophagy, in metabolic control, in proteostasis, and organelle dynamics. So the other protein which which is talked about in this paper, like MFN2, you can see here. So what is exactly this MFN2 is? MFN2s are dynamine like guanosine triphosphatase, or they are especially the GTPases. They use GTP as an energy source and responsible for the mitochondrial fusion, and which is a fundamental event to fuse to mitochondria and that enhance the OXPHOS activity, the capacity of the mitochondria. It is also reported that these MFN2 also help tethering the mitochondria to the ER, and they are present on ER especially. So people have in this paper which was came in 2008, they have shown that these mitofushin 2 are present on ER, and they interact with the MFN2 or MFN1 which are present on mitochondria, and they tether the mitochondria to the ER, which facilitate this interaction. And if you talk about the SERCA, which is a protein and the key regulator of cellular calcium homeostasis, also the name came from sarcoendoplasmic reticulum calcium ATPase, SERCA, pumps acts to transport calcium ions from the cytosol back to sarcoplasmic reticulum. So basically, they are discovered, I think, in uh, muscle cells. That's why this name came from. Now, the second term which is used in the title is what is the T cell metabolic fitness, basically. So this is a representation. This is a cartoon which is taken from this reference, which is given here. So I added these references and these cartoons to simplify, to make it understandable. So if you see, for example, knife cells, if you compare the total metabolism of knife cells, whether it is more glycolytic or oxphos, we can say. So you can see here knife cells have more of oxphos metabolism and less of glycolysis in these cells. When these cells activate, the oxphos is more or less, or you can say little decreased, but most prominently we can say there is an increase in glycolysis when cells are activated, and then these cells go for different fate, which is depend on the uh, status, physiological status, or what we can say the microenvironment in which these cells are activated. Either they will be exhausted after some time, or they will become memory cells. If they are exhausted, there will be less oxphos, and if the intervention, like uh, they want to maintain this effector function, they have to maintain the glycolysis and oxphos comparatively. So what we can say here is, for the function of T cells at a particular stage of their differentiation from naive to the activated to the exhausted or whatever the memory cells, they have to maintain a specific balance between oxphos and glycolysis. Sometimes they are more lean to the oxphos, and sometimes they are more lean to glycolysis. And it has been observed that in different stages of differentiation of these cells, they have different balance of these glycolysis and oxphos in these cells. Similarly, in this paper, we will see these markers which has been investigated for the purpose to show that whether these cells are activated or these cells are exhausted. Especially the marker you can see here, TIM3 and LAG3 and PD1. These marker has been used in this paper to show whether these cells which they are studying are they activated or exhausted. And it has been known that the aberrant mitochondrial phenotype, like reduction in the total mass and reactive oxygen species level, and increased depolarization and fragmentation of the morphology of the mitochondria, or hallmark of exhausted CD8 tumor infiltrating lymphocyte, we can say. So with this knowledge, we will move forward. In this paper, they have given various references to make a point that the nature of mitochondrial behavior in tumor infiltrating T cells remain poorly understood. That's why they have done this research. So the behavior of these T cells and how the metabolism in these T cells is happening, how it is associated with the function of these T cells is poorly understood. People have done they have done extensive research by using these T cells, especially CD8 T cells, and also done the research using these NK cells, and they have shown the role of these NK cells in immunosurveillance and so on. So let's come to the first figure in this paper, and what it is shown. In the first figure, what they try to show is they have used this renal cell carcinoma patient samples, and they did this IHC, and they also correlated the expression of this molecule MFN2 mitofugin 2 to the overall survival of these patients. So what they have found is increased expression of MFN2 is correlated with the better survival compared to the low expression of this MFN2. What they did is they also did this IHC, in which they stand for CD8 tumor infiltrating T cells and MFN2 expression, and what they found is the samples in which high MFN2 is expressing in CD8 T cells, these patients have overall survival more compared to the patients which have low MFN2 in their CD8 T tumor infiltrating lymphocytes TILs. Similarly, the disease-free survival also correlated with the high MFN2 expressing CD8 T cells. So if patients in patient tumor samples, if the expression of MFN2 is more in CD8 T cells, there is more disease-free survival in these patients also. Furthermore, what they did is they also found that this MFN2 is, when the MFN2 is high, the infiltration in the tumor is more of these CD8 T cells compared to the CD8 T cells which have less MFN2. We suggest that maybe this survival is related to the this presence of these CD8 T cells in the tumor. If more CD8 T cells are present in the tumor, more regression of tumor will happen and the survival will increase. Similarly, what they have found is they checked for different markers of activation of CD8 T cells like PD1 double negative and TIM negative, which suggest naive T cells, and PD1 positive and TIM negative, which is a marker which suggests the activated T cells, and PD1 positive and TIM positive, which suggest exhausted T cells. And they probed for the expression of MFN2, and what they found is when the T cells are activated, MFN2 expression increases, and when the T cells are exhausted, the MFN2 expression is decreasing comparatively. So they correlated this expression of MFN2 with the activation of CD8 T cells. Furthermore, what they did is they isolated these CD8 T cells from patient samples, and they did single cell RNA sequencing, and they found there are 5 major clusters, and from cluster 1 they are going to separate different nodes. Are, you can see here, different nodes: C5, C4, C2, and C1 is here already, and C3. So they make a pseudo time graph from early to the late. So this is the early and these are the late CD8 T cells subtypes which they plotted here. Further, they also make this UMAP, in which they have shown these different clusters of CD8 T cells which were isolated and sequenced in the renal cell carcinoma samples. They isolated 3569 single CD8 T cells, and they did the single cell RNA-seq. So here they plotted different markers, like C1 is LEF naive cells. They are expressing LEF1, which suggests they are naive cells, which is here, and Granzyme, they using the Granzyme marker as a pre-effector cells, and CX3 CR1 as a cytotoxic effector cells, which is mostly here, and the exhausted CD8 T cells which are expressing LEG3, which is here, you can see, and Ki67, which is a proliferating marker which has been shown here. So these are the overall clusters which they have found, and they use this data and they plotted it in a pseudo time analysis, this graph in which y-axis is showing the expression level of CCR7 and how with the pseudo time it is increasing or decreasing. Similarly, y-axis is showing expression of different like granzyme, hyphen gamma, and different MIK Ki67 basically, and LAG3, and they correlated this expression with the MFN2 expression. You can see here there is a decrease in CCR7 which suggests that the naive cells are decreasing. Activation hyphen gamma is going up from this cluster 1 to the cluster 5, especially you can see here the color coding, cluster 1 to the cluster 5, and the cluster 1 to the cluster 5, if you see the CCR7 is decreasing. Similarly, the KI-MI67 is increasing if we go, and if we go from cluster 1 to the cluster 5, we can see that in pseudotime analysis there is an increase in MFN2 expression. Similarly, the T cell activation score is also increasing, which suggests that the MFN2 expression is correlated with the T cell activation. As the T cells are activating in this course, there is more expression of MFN2. Similarly, oxidative phosphorylation score is also increasing, which suggests that this MFN2, because it is already known that MFN2 expression correlate with the increased OXPHOS capacity. So these OXPHOS related genes are also increasing, and which is correlated in the pseudotime analysis. Similarly, the allograft rejection score, which is calculated by the expression of gene pattern, which suggests that the activation of T cell basically, and the tumor basically working as an allograft, you can say. So if the allograft rejection score is increasing, it means that these T cells are able to kill these tumor cells, or we can say the this activation of allograft rejection type of activation of T cells is happening. So overall, what it suggests that this increased MFN2 expression is correlated with the increased effector function. So let's move further. Further, they wanted to know what exactly controlling this expression of MFN2. So they used different in situ, or what to say, the tools, softwares we can say, on this RNA sequencing data to find out different transcription factor which are probably involved in the expression of this MFN2. And what they found is, by analyzing this data, they found that AP1 could be one of the transcription factor which may be responsible for the increased expression in this activation of T cells. So it is already known that AP1 can influence the expression of MFN2. Furthermore, they also checked for the binding site on MFN2 promoter, and they found that there is a AP1 binding site is there. Further, they did a luciferase reporter assay in which they use AP1 directly. You can see here they use AP1 directly in MFN1, as well as they use AP1 directly to the MFN2 to see the promoter activity, and they found there is increased promoter activity when they have used AP1 uh, and in both MFN1 and MFN2 promoter, but the increase in the promoter activity is more in MFN2, which suggests that mostly this increase in AP1 is increasing the expression of MFN2 in these cells. So next what they did is they just knocked out this MFN2 from these CD8 T cells. So basically they knocked it out in both because this MFN2 flox CD4 Cree and these mice, they knocked out this MFN2 in both in CD4 as well as in CD8. But when they have checked it in knockout mice, there is an increase in the tumor growth when they knocked out CD8 and CD4, the expression of MFN2 in both CD8 and CD4 here. So there is an increase in the tumor growth, which suggests that either CD4 or CD8, either both or single cells, immune cells are involved in this tumor regression basically. They also checked for the expression of IFN gamma, which is a T cell activation marker, and they have seen that in these mice there is a decrease in IFN gamma expressing cells, or the percentage of these cells basically. Furthermore, what they did is they also see the percentage of IFN gamma per CD8 T cell percentage, and that is there is also decrease in that IFN gamma in these cells, in knockout mice basically. Ki-67 is also decreasing, which suggesting that cells are not much proliferating in these CD8 T cells are not proliferating in knockout mice compared to these mice. Similarly, they also have seen their annexin 5 positive CD8 T cells in this tumor infiltrating this tumor microenvironment, and which is suggesting there that these cells are dying. So basically, when CD8 T cells, especially these tumor infiltrating CD8 T cells, they die if, for example, the activation happens and they exhausted, and after they are exhausted, they will die. So here we can see annexin 5, which is a marker for the apoptosis, and they are showing that more annexin 5 positive CD8 T cells are there, which means more CD8 T cells are dying. To further correlate this with the CD8, what they did is they use anti-CD8 in both knockout and wild type, and they see there is the difference between this tumor is gone when they use CD8 cells, CD8 anti-CD8 basically, which suggests that mostly CD8 cells are involved, not CD4 cells are involved in this tumor increase after knockout. Similarly, they also checked the effect of this MFN2 knockout on the anti-PD1 therapy, and they found that in wild type, you can see wild type is here, and when they given this anti-PD1 to the wild type, that decrease in the tumor you can see here, so it is working fine. But you can see in knockout, when they have given, the tumor volume is high, and when they given anti-PD1 therapy, it is not working in this tumor, which suggesting that the ablation of MFN2 increases the tumor progression because of these non-functional or reduced function of the CD8 T cells, and also this have impact on this anti-PD1 therapy. Further, they isolated CD8 T cells from these tumors, and they did the same analysis which they have done with the CD8 T cells which they were isolated from human samples, and they found a similar trend in the MFN2 high and MFN2 low, uh, as they found in MFN2 high and MFN2 low tumors. They found in the knockout, similarly, uh, the activation markers are decreasing basically. So, for example, if you see C5, so C5 is in this yellow, it is decreasing compared in knockout compared to wild type. Similarly, if you see naive are increasing in knockout mice compared to uh, this wild type mice. Similarly, the proliferating cells are also decreasing in knockout compared to the wild type. So overall, this data is suggesting that in the MFN2 knockout, especially in CD8 and CD4 T cells, is decreasing the activation of CD8 T cells in this microenvironment. So overall, they concluded that the MFN2 support the long-lasting activation, proliferation, and functionality and survival of T cells within the tumor microenvironment. Furthermore, in the similar experiment, similar marker they have used different marker to show that in knockout there is a less granzyme P positive CD8 T cells. There is more KLRG1 percentage of knockout percentage in the knockout, and there is a more CTLA4 positive CD8 T cells in the knockout. Similar other marker also they have used, and overall they have shown that genetic ablation of MFN2 in CD8 T cell decreases the effector function of these CD8 T cells. Further, so the next experiment they wanted to check the effect of these uh, MFN2 knockout and the MFN2 expression on the OXPHOS activity. They want to correlate it with the mitochondrial function. What will happen if the MFN is knockout, and what will happen if the MFN is wild type to the mitochondrial function. So to understand the further experiment, I have added this introduction here. For example, here we can see. So if we give directly fatty acids in the microenvironment or to the cells, they can utilize these fatty acids using this mitochondrial OXPHOS, or we can say mitochondrial function. So these fatty acids are transported inside the cells, and then these fatty acids gone through this beta oxidation, and the energy will be generated throughout this. So this particular transporter which transport fatty acid inside the mitochondria, it is used in this study. They inhibited it to show that the mitochondria are involved. Furthermore, to synthesize these fatty acids inside the cells, these enzymes are involved. For example, SEC and FASN, and these enzymes are involved in the synthesis of fatty acid inside the cells. So these markers are also used in the further experiments. Similarly, they have used seahorse experiment, or we can say the oxygen consumption rate here to show the functionality of mitochondria. So it's a basic diagram of what will happen, what is happening, like how we can say something. So for example, this oxygen consumption rate is representing the basal respiration. When you give oligomycin inhibit the complex 4. So by inhibiting complex 4, overall most of the before the complex 4, all these complexes are not able to give this electron to complex 4, so the overall oxygen consumption will go down. So it will go down, we can see here. So oligomycin given, and the oxygen consumption rate goes down. Further, when they added FCCP, FCCP will depolarize the mitochondria, so the mitochondria will use their maximum capacity to re-establish this proton gradient, and so this will show as the maximal respiratory capacity. So mitochondria use their maximum potential to restore this proton gradient, and when you give the rotenone, which is a complex first inhibitor directly, and then antimycin E, which inhibit complex first, and the cell will overall goes down. And the difference between this basal respiration and this maximal peak of the maximal is the reserve capacity, so which suggests that uh, this capacity suggests how much mitochondria have reserve function. When the nutrients are available, they can use this in that time. Let's see further. So in this data, so they use this MFN to knockout versus the wild type, and they try to find out what are the genes which are differentially regulated. They found that the genes which are related to the mitochondrial organization, generation of precursor and metabolic and energy related genes, antigen processing and presentation related genes, these genes are in this case are altered. They found there is a down regulation of interferon gamma response, which we have seen that effector function is goes down in MFN knockout, the CD8 T and CD8 T cells. Glycolysis is also goes down, fatty acid metabolism goes down, and oxidative phosphorylation is goes down. and adipogenesis also goes down, which is kind of representing the in uh, uh, the de novo synthesis of these fatty acids inside the cells. They use these markers like FASN, and which we have talked about later, the CPT1, which is a marker to transport the fatty acid inside the mitochondria, which is also going down, and this EKK, these genes, all genes which have been shown here are involved in the de novo synthesis of these fatty acids inside the cells. So all these things are goes down, and also CPT1 is also going down. So these fatty acids which are available cannot go much inside the mitochondria in knockout. So overall, this suggests that loss of OFN2 impair the mitochondrial respiration capacity of intratumoral CD8 T cells fueled by fatty acid oxidation. Further, what they did is they did this OCR, check for the oxygen consumption rate, and you can see here in the knockout the oxygen consumption rate, the basal oxygen consumption rate is decreased, which means that without any further interference with these chemicals, the basal is also low, which suggests that the total mitochondrial, or we can say the mitochondrial function, is down in MFN2 knockout CD8 T cells. And the maximal oxygen consumption rate, the difference you can see, the reserve you can see here. If you see the reserve capacity of mitochondria, it is very less in the knockout, but in the wild type it is quite good. So basal oxygen consumption rate is goes down, the maximal oxygen consumption rate is also goes down. They use this body pi 500 this dye to show the total lipid content of the CD8 T cells, which is also going down, which also correlate with the gene expression. And further, what they did is they did one more experiment here. So we will visit again these graphs. So these graphs are, so what they did is they use this palmitate. So what palmitate do is palmitate enhances the mitochondrial oxphos. So they added palmitate in the wild type and the knockout. So what, and they also use this ETX, which is an inhibitor of CPT1. It means that if the palmitate is given and the ETX is also given, this palmitate cannot go inside the mitochondria, so it cannot increase the oxphos in this case. So if you see, this is a wild type. When the in the wild type, when the this inhibitor of CPT1 was given, the maximum and the basal oxygen consumption rate is goes down. Here you can see here it goes down, and it comes compared in the compared to the knockout. You can see the knockout and the ETX treated cells, they have almost comparable oxygen consumption rate as well, basal consumption as well as maximal oxygen consumption rate. So we suggest that in knockout they are not able to use this palmitate efficiently. It means the oxidative phosphorylation is compromised in these cells. And further, when they use ETX, it goes further down, which suggests that that's the same conclusion we can make. Similarly, they use 2-NBDG, which is a glucose analog, fluorescent glucose analog. So this 2-NBDG suggests that there is a decrease in glycolysis also, but it is not very much decrease compared to the oxphos. So glycolysis is also is decreased, but the oxphos is more aberrantly decreased in this case. Same thing, it has been shown here. You can see here relatively the body pi MFI, which suggests that total overall lipid accumulation is decreased in these CD8 T cells. So when they given 2DG, which is a 2-deoxyglucose, which basically inhibit glycolysis, you can say. So when the glycolysis is inhibited in this wild type, there is a decrease in the total lipid content, and when the CPT was given, there is a drastic decrease in this lipid content. In the knockout here, the basal level of this, or we can say the total in the knockout is already decreased, but when the 2DZ was given, it means the glycolysis is inhibited, it goes more down, and when the ETX was given, it also goes down compared to the wild type, it is less. Similarly, this graph is directly similar to the IF-1 gamma in CD8 T cells in these cells, which suggests that uh, these activation of CD8 T cells is correlated with the OXPHOS capacity of these knockout cells. It means that if they have less OXPHOS capacity, they will produce less IF-1 gamma or less activated CD8 T cells, and which is also correlated with the infiltration and everything we can think about, which has been shown in this paper. Further, so now the question they were asking how this is happening, basically how this MFN2 is able to, the knocking out this MFN2 is able to curb this mitochondrial function, how exactly it is happening. So to do that, what they did is they use spleen and intratumoral CD8 T cells isolated from wild type and MFN2 knockout B16 bearing mice, and they try to see what is happening with this mitochondria and ER contact. So basically MFN2, as we recently talked in the previous slides, that MFN2 is responsible for the tethering of the mitochondria to the ER. So they try to figure out what is happening to this ER mitocontact in this tumor and spleen. So if you see here, first they checked for the mitochondrial morphologies, and you can see this is spleen, this is tumor, this is wild type, and this is spleen, this is tumor. So if you compare the tumor with the tumor, so not much difference in the mitochondrial morphologies is there, but when you compare the Mendel's coefficient basically shows how much these two molecules are co-localized. For example, in this case they use COX4, which is a complex 4 of ETC, electron transport chain, and the kalinexin, which is a marker for the ER. So how much they are co-localizing. So in the case of tumor, you can see compared to spleen, they are more co-localizing in the tumor. But when you knocked out MFN2, this difference is gone. It means that there is a less mitochondria ER contact in the tumor uh, in knockout MFN knockout cells, which is they also did by electron microscopy. And sorry. So further, what they have concluded is the MFN2 mediated mito-ER contact and mitochondrial calcium influx are necessary for superior mitochondrial metabolism and effector function of CD8 T cells. So although this conclusion is not directly can be made by these experiments, but they have did more experiments which we are talked about. We will talk recent in the upcoming slides. Basically, this data is showing that after knocking out MFN2, there is a less connection between mitochondria to the ER. Further, so in knockout, obviously the percentage of mitochondria to ER is decreasing. Here also you can see what they have done here is they use two different type of lysates. One is the mitochondria ER junction lysate, and other is whole cell lysate. So when the mitochondria ER junction lysate were used, you can see the kalinexin, the knockout is less, and the although the complex 4 expression is almost comparable, MFN2 is less, obviously because this is knockout, MFN2 is not there. So in mitochondria ER junction, there is a less kalinexin. It means there are less interaction between ER and the mitochondria, which was further quantitated here. Further, what they did is they also used this dye road 2, which suggests the total calcium intake, because it has been known that when the ER and mitochondria contact happen, the transport of calcium in both sided is happening. So they wanted to know what is happening with the calcium concentration in these the mitochondria of these cells. So what they have found is you can see here is spleen, the total road accumulation, which indirectly suggests the calcium accumulation. In the knockout tumor, it is very less compared to the wild type. Even so, even the in the wild type spleen and the both tumor the total calcium uptake is decreased in the knockout compared to the wild type, which is quantitated here. You can see here in spleen, compared to spleen in tumor, there is a more calcium in the mitochondria, but when you knocked out MFN1, sorry, MFN2, there is no difference between spleen and tumor, which suggested there is a decrease in the total overall calcium in the mitochondria. And similarly, they used this RU360, which is an calcium uptake inhibitor in the mitochondria. So when you inhibit the calcium uptake inside the mitochondria, there is also a decrease in the body pipe, which means the total lipid content of the cells is also decreased here. And the IFN gamma is also is decreasing when you give this inhibitor of mitochondrial calcium uptake. It means that what they try to correlate here is that this MFN2 knockout is creating a situation in which less mitochondria ER junctions are happening, which in case decreasing the total calcium amount of mitochondria, and the decrease in this total calcium amount in the mitochondria is decreasing total this IFN gamma expression in the CD8 T cells, or we can say effector function of these CD8 T cells in these intratumoral CD8 T cells, which were isolated from the B16 tumor bearing mice. Further, what they did is they try to establish this mitochondria-ER and this calcium is responsible for this CD8 T cell function. So MFN2 mediated Mito-ER content contribute to the enhanced mitochondrial metabolism and effector function. So basically they are correlating this amount of calcium inside the mitochondria to the effector function of CD8 T cells. Here also what they used CD8 T cells isolated from peripheral blood and tumor tissues of renal cell carcinoma. So previously they used the CD8 T cells from the what they did is they wanted to know what are the partners of this MFN2 protein. To know that, what they did is they, they did this they checked for the expression of this MFN2 in the cytoplasm, ER, and the mitochondrial lysates of CD8 T cells, and they found that in the ER associated lysate they did not find the expression of MFN2, although there was the expression of kalinexin, which suggests that this is the ER lysate. Similarly, so this data made them to think whether this MFN2 is actually expressing in ER or not. So in this paper they have included that they are not explicitly saying that it is not present on ER, the MFN2, because it is already reported that it is present on this ER membrane. They are saying that maybe it is it is only present where the mitochondria and ER connection or this tethering is happening. Maybe this MFN2 is only present in that part of the ER, not in all the ER fractions. So further, what they wanted to know is what are the partners which are binding with this MFN2. To know that, what they did is they used this MFN2 plasmid, and they made these competent cells, and then they transfected it in SFN9 cells. From there they purified this MFN2, and because this MFN2 is flag tagged, and then this purified MFN2 is incubated with the cell lysates. So they used two different cell lysates. One is HEK293 T cells and other cell lysate from T cells. They wanted to know what are the other proteins which interact with this MFN2. So after incubating this cell lysate overnight with this purified MFN2 antibody, they did mass spectrometry analysis, and they found that this MFN2 is interacting with MFN2. Obviously, it is already known MFN2 is interacting with MFN2, but also it has been revealed to them that it is also interacting with SERCA2, SERCA1 and SERCA3. So out of these three isoforms of SERCA, they choose SERCA2 because it has higher expression in human tissues. Further, this data is showing that this is the mass spec data of the HEK293 and the CD8 T3 cell, CD8 T cells. Furthermore, to confirm that this MFN2 is interacting with SERGA2, they did this co-IP and this co-IP experiments, in which what they did is they use HEK293 T cells, and for example here they did IP with HA. HA is SERCA, so it is present in the this SERCA 2 expressing HEK293 T cells. It is here, we can see here. It is also present here with MFN2 and SERCA2 co-expressing cells, and the band is bigger. It means that they are interacting. The other thing they did, for the flag, when they did the IP, the flag, the MFN2 is flag tagged. So here we can see here, for example, at the same band which was previously seen, and they also seen with the SERCA2, and in MFN2, when obviously here we can see MFN2 flag is there, so MFN2 band is there, and in flag MFN and the SERCA both co-expressing cell, there is a bigger band, which suggesting that they are co-expressing. So this serum can be used to investigate the co-expression, and when they did the input means the HEK293 T cells. These are the isolated protein markers. So when they did this with HEK293 T cell lysate, you can see here the flag and HA SERCA, they're both similar band. For example, these bands are replicated here and this band is replicated here, which suggesting that they are interacting in HEK293 T cells. Similar experiment they did with the T cells, and they found that SERCA2 and MFN2 is interacting in the T cell also. And they also validated this with a confocal analysis in the Jurkat cells, and they found that this interaction is happening in Jurkat cell also. So overall, this is suggesting that the SERCA and MFN2 are interacting in HEK293 human T cells, Jurkat cell, and cultured human CD8 T cells. Further, to investigate this interaction and they validate these results that they are interacting, what they did is they took these crude mitochondrial fractions in which the mitochondrial and ER junction is there, and they just stained it with the MFN2 and SERCA2, and these mitochondrial crude isolated from anti-CD3 CD28 activated human CD8 T cells. So we can see here there is a co-localization of MFN2 and SERCA2, which is suggesting that they are interacting. This is the direct evidence for that. Furthermore, what they did is they used this proximity analysis in which if two proteins are close enough, they will give this red dot, which suggests that if this red fluorescent is observed, it means that these two proteins are close enough. You can read about this particular method, how they did it. So basically this method utilizes two antibodies for two different antibodies for the different proteins. Then these antibodies have some tagged oligomers with them, and these oligomers then used by these polymerases, and they form a longer nucleic acid chain, and this nucleic acid is can be tagged further. So in this experiment, basically if two proteins are close enough, then only this red dot can be seen in this case. So here we can see here MFN2 and SERCA2 are interacting. Furthermore, they did this analysis in mitochondrial ER junction and whole cell lysate. So you can see here when they use this SH circa 2, it means that circa 2 is downregulated, and when the circa 2 is downregulated, mitochondrial ER junction is also, the expression is downregulated. It means that circa 2 is facilitating these mitochondria ER junctions. Further, in wholesale light there is no difference, obviously. Furthermore, they also use SF9 insect cells, and they purified these circa 2 histake, and they did this silver staining and comesci staining, which is suggesting that these two proteins are interacting again. Furthermore, this proximity analysis is also done in freshly sorted CD8 T cells from patient with renal cell carcinoma. So they also checked this proximity ligation assay with this proximity ligation assay in human, the in vitro cultured CD8 T cells, as well as in the isolated CD8 T cells from patients. Now, the question they were asking, exactly what part of the this MFN2 structure or this protein is interacting. So here we can see this is the protein structure of MFN2, and there are various regions which are involved in different interactions. Especially, for example, if you can see this R59A or this site, so they did the point mutation, or to know the role of this particular site. So this is showing the point mutation. So this particular region is responsible for homodimeric interface. This particular region is responsible for GTP binding and hydrolysis, and this region is responsible for hinge. This is a hinge region, and so now what they did is they use these different variants of MFN2 in which the point mutation is there. So basically, this, if point mutation is there, that particular part is not functional. So if you see in these three point mutated variants, there is no interaction between SERCA2 and MFN2, but there is an interaction when this particular point mutation, which is R250A, is done, but still there is interaction, which means that this particular site is not involved in the function of interaction between SERCA and MFN2, and this particular site R59A is responsible for the MFN2 homodimerization. So it means that all the other sites are responsible for the function of the MFN2, especially the GTP hydrolysis and everything. All these three variants are required for the function of MFN2. So overall, what they suggested is the intact GTPase machinery and conformational flexibility of MFN2, but note its homodimerization ability are indispensable for interaction with SERCA2. So by doing this experiment, they got to know where exactly the SERCA2 is interacting with MFN2. Furthermore, to substantiate this result that they are interacting, what they did is they used this this assay in which they directly use this purified SERCA2 and MFN2, and they wanted to know what exactly will happen with the utilization of the ATP in this case. So because we know that the SERCA2 is also ATPase, so their activity can be measured by using the use of the ATP. So when they added different concentration of MFN2 to the SERCA2, they found that there is a substantial increase in the use of the ATP. It means that if you are adding more MFN2, there will be more activity of SERCA2 in this case, which is also shown here, the same data. And furthermore, they use a constant concentration of SERCA2, and then they added various variants of this MFN2, and they found that these particular variant, for example, this R94Q, which is responsible for the activity of MFN2, when they added this one, there is no change compared to the control or the single SERCA2 activity. But when they added wild type, there is an increase, and but and when they have added this R59A, there was an increase, which is this variant have actually no effect on function, so that's why this activity is comparable to the wild type. This is also further analyzed here. This data is represented here. In this experiment, to substantiate this or validate this data more, they use these giant unilaminar vesicles. This data. So what we can do here? So these vesicles can be fused with smaller vesicles which are containing these molecules. For example, in this case, the SERCA2 is here. They're fused, so the SERCA2 will be distributed on the surface of this membrane. So basically, this experiment is they try to do it in a tube. So they are creating an ER and then they are giving this MFN2 and they are seeing whether they are interacting. So here, when you can see here, when the SERCA2 was fused with these giant laminar vesicles, there is an increase in calcium inside these vesicles, which is suggesting that the SERCA2 is working fine. And then they added the MFN2, and they've seen that there are more calcium going inside, which is suggesting that the MFN2 and SERCA2 interaction basically increasing the activity of SERCA2, so the more calcium can be taken up by these vesicles. This data is also validated by using various isoforms of MFN2 or point mutated these variants, in which they have found similar data that the this hinge region basically which have no effect have increased calcium uptake. Wild type have increased calcium uptake, but when the this non-functional MFN2 basically added, there is a not a very much comparable to wild type. There's a calcium accumulation inside these vesicles. So overall, what this data is suggesting that SERCA2 is interacting with the MFN2, and this interaction is increasing the activity of SERCA2, which in result increases the calcium uptake. But now what is the next step to do? As we know that SERCA2 is present on the ER, and when the MFN2 of mitochondria is interacting with it, it means that there are more calcium will be taken up by the ER. So to do the further analysis, what they did, a very interesting experiment they did, in which they used these linkers. So what exactly this linker do? So when they express these linkers inside the cells, so these linkers will increase the tethering of mitochondria to the ER. So there will be more mitochondria ER junctions comparatively. So for example, here you can see basically here they also checked for this fluoro5F, which is suggesting the calcium uptake in different variants. Now come to this data. So when they use this knockout CD8 T cells, and so they have seen that there is a decrease in the SERCA2 activity, which is suggesting that it is not only in vitro, it is basically not only the outside the cells they have shown previously, but it is also happening inside the cells. The end result is that SERCA2 activity is going down basically in the CD8 T cells. Furthermore, what they did is they use these linkers. So for example, you can see here when they use linker, there is more cholinesterase in these mitochondrial ER junction lysates, which means that these linkers are increasing the interaction between mitochondria and ER. You can see here in knockout the interaction is less. When the linker is used, it is restored again. So what will happen? They checked for the calcium uptake by the mitochondria. So by using this technique, freight ratio, in control it is here, but when the linker is given, there is more interaction. It means then there is more transport of calcium to the mitochondria. In knockout, there is a less calcium uptake, but when the linker is given, there is a substantial increase in the calcium inside the mitochondria, which is more than the wild type when the linker is used the wild type. That was a very interesting data. So which is suggesting that this interaction of SERCA2 to MFN2, it is not unidirectional. It is not taking this calcium from the mitochondria to the this ER, but it is like it is making, it is try to do this homeostasis of the calcium. For example, here by the cartoon they want to show here, for example, when the SERCA2 and MFN2 is interacting, the calcium is going more in the mitochondria. When the calcium goes more inside the mitochondria, just because two mitochondria and this ER is there, so there will be more transport of calcium from the ER to the mitochondria. But when this SERCA2 and this MFN2 are functional, so MFN2 will increase the activity of this SERCA2, so the calcium which could have gone more inside the mitochondria will be taken back inside the ER. So by doing that, they are basically maintaining the homeostasis. So if the MFN2 is working, for example in this case it is not working, so that's why the uptake of calcium inside the mitochondria is way more than compared to the wild type with the linker. So because the SERCA2 activity is compromised here in this case, because this MFN2 is knockout in these cells. So they also checked for the apoptosis rate with the linker, because it is known that if, for example, if more calcium is taken up by the mitochondria, but there is a threshold. If the mitochondria cross that threshold, the apoptosis will be started, because mitochondria cannot handle that much calcium and the cell eventually will die. So in this case you can see when the linker was given to the knockout, there was a unidirectional flow of calcium to the mitochondria, which in result do the apoptosis of these cells. Similarly, to know the effect of this calcium transport on the uh, tumor, or the indirectly the effect of these CD8 T cells on the tumor. So what they did is, for example, so they adaptively transported, they adaptively injected these knockout CD8 T cells. For example, here in the wild type, there is a linker. So there is a basically, if the linker is there, there is a decrease in the tumor in the wild type, because more calcium will go, more mitochondrial oxidative phosphorylation will happen, and the more CD8 T cells will be activated in this case. But when the linker in the control and the CD8 knockout, for example, you can see in the linker also they have shown that there is a significant decrease in this case also. But it is not very much a reversal, which is showing that the homeostasis of the calcium is more important than the unidirectional transport of the calcium inside the mitochondria. So further to check this calcium flux, or to establish the function of this calcium flux in this anti-tumoral immunity of intratumoral CD8 T cells, what they did is they used this MFN2 flux log CD4 and they crossed it with the OT1 mice, and then they make OT1 MFN2 flux log CD4 mice. Out of that mice they isolated the CD8 T cells, and they used this lentiviral to transfect different variants, and then they injected these cells to the B16 OVA mice. Further, so we can see here this data here. So when the wild type MFN2 is expressing CD8 T cells were injected in the control, versus the knockout CD8 T cells were injected. So here we can see the this is the control with which the wild type MFN2 is there. But when the knockout is expressed, there was an increase in the tumor. The red you can see here, there's an increase in the tumor compared to the wild type MFN2. When the with the this double negative MFN2 mice were transfected with the wild type MFN2, for example, were added with the wild type MFN2. So it is compared to the this thing. So basically compared to the wild type mice, basically when you injected the wild type MFN2. But when you use this variant, which is not responsible for this MFN2 SERCA activity, so it is kind of working as a wild type. So there is a increase in the tumor. But when they transfected the cells which are expressing this particular R94Q variant, which is non-functional variant of MFN2. So when the MFN2 knockout mice were adaptively transferred with the CD8 T cells which are expressing R94Q, which is a non-functional MFN2 variant, they shown there is an increase in the tumor, which is suggesting overall this data is suggesting that uh, this the same data which they have shown in vitro, they have now shown it in vivo also, that it is working like as it was working in vivo, in vitro, sorry. In this case also they use this MF1 knockout OT1 TCR transgenic mice, and you can see here in control, in wild type, and the functional, everywhere the mitochondria-ER junction is fine. But when this non-functional was transfected in this MF1 to knockout mice, there was less mitochondria to ER interaction in this case. So this control is MF1 to knockout, and then the expresses. So you can see here MF1 2 is not there. It means that this is the knockout for MF1 2, and when they express different variants, the MF1 2 was expressing, but MF1 in this particular variant, the ER and mitochondria junction is not happening. Similarly, in these variant, they also checked for the IF1 gamma expression, which is suggesting how much the CD8 T cells are activated. So in this case, for example, it is like 26.3%, but when, because this is wild type MF1 2 mice and this is the control one. Now when this knockout was expressed with the control, control is like MF1 2 double negative, it means the double negative. So the mice have this CD8 cells which are double negative for MF1 2 were injected in these cells. So there's a decrease in IF1 gamma. But when the wild type was expressed, the wild type of MF1 2 was expressed, there was a restoration of this IF1 gamma. And then when this functional variant of MF2 was expressed, then there also this thing sustains. But when this particular variant was expressed, this variant is non-functional because this interfered with this GTP lysis activity of MF2. So we can see there is a decrease in the IF1 gamma also. So they correlated this with the CD8 T cell function also. Furthermore, this is the basically they did this quantification of these blocks here. Furthermore, what they did is they wanted to know these calcium flux are provided for antitumoral immunity for intratumoral CD8 T cells. So further they check for IF1 gamma, so it is going down. They checked for Mendel's coefficient, which means that there will be less interaction. So it is kind of the data which were expected in this, because the individual data they already have shown previously, but now they are doing with the different variants. In this particular experiment, what they did is they used this mitochondrial calcium uniporter. So they expressed this mitochondrial calcium uniporter, which so what does it mean is the calcium uptake will be increased because mitochondria in this these particular CD8 T cells will have this expression of this MCU. So you can see here, so this is the control highest tumor in which MFN2 is knockout. But when this mitochondrial calcium uniporter was expressed, this there is a change in the tumor growth, or we can say there is a decrease in the tumor growth compared to this wild type MFN2, which is suggesting that this calcium inside the mitochondria is responsible for the decreasing the tumor volume in the CD8 T cells. Furthermore, in the MCU, only MCU expressing CD8 T cells, there is an increase in IF1 gamma compared to the MFN2, although it is low, but there is an increase. Then further, this MCU2 expressing have a relatively higher ROD2 MFI, which is suggesting there is more calcium, and this bio body pi, which is suggesting more fatty or the lipids inside these cells, basically when they are expressing this MCU on CD8 T cells, which means that if this calcium uniporter even alone is expressed expressed in the CD8 T cells, there will be an increase in IF-1 gamma function, and there is an increase in the calcium uptake inside the mitochondria. Obviously, that's why they are expressing this MCU, and there is an increase in the fatty acid or the lipids inside these cells. So now to understand this calcium business, how this calcium is working, what will happen when the more calcium go inside the mitochondria or less calcium go inside the mitochondria. So I put this cartoon here, or this, so what we can see here. So VDAC is responsible mainly, and this MCU in the inner mitochondrial membrane allow the calcium to come inside the mitochondrial matrix. So if the calcium is very high in the concentration in the mitochondria, there will be opening of this MPTP, and subsequently this release of cytochrome C, and there will cell will die, which we have seen in the case of the linkers, when the cell were dying more because the more calcium was flowing unidirectionally. Now in this case, for example, these calcium is responsible for the activation of different enzymes, especially which are involved in this TCA cycle. For example, this PDH phosphatase, isosaccharic dehydrogenase, and oxoglutarate dehydrogenase. So there are various mechanisms by which this calcium activate and these enzymes, which is shown here. So overall, what he's suggesting is when the calcium is more inside the mitochondria, there will be more activation of these TCase associated enzymes, and then there will be a more increase in the OXPOS activity of these particular cells. And as it has been shown that if there is a more calcium, and the reason there is a more calcium because there is more mitochondria and ER tethering is happening because of SERCA and this MFN2 interaction. So further, what they did is they wanted to check the effect of this MFN2 on the cancer immunotherapy. So what they did exactly here. So in this case, what they did is they used this conditioned media. So what they figured out is the media in which they are culturing these CD8 T cells, for example, is little bit different in the microenvironment of these tumors. So they try to create a this tumor microenvironment like media, which they called a conditioned media. So when they cultured in this normal media these cells, so they found there is a basically when they cultured in this conditioned media, there is an increase in this relative kalinexin, which means there are more interaction when they come when they culturing is these cells in this conditioned media. The oxygen consumption rate is high, which is suggesting that these conditioned media are supporting this OXPOS function of these CD8 T cells. Furthermore, obviously there is a correlation with the IF-1 gamma and the oxygen consumption rate in this case. So there is an increase in IF-1 gamma because there is increase in the mitochondrial OXPOS. Further, even the viability of the cells are also more. And furthermore, what they did here, for example, so how they did these experiments. In this case, they isolated the tumor, then they make these tumor lysates. These tumor lysates were used to stimulate these dendritic cells. Similarly, these dendritic cells were previously isolated from the blood, and then these dendritic cells were co-cultured with the CD8 T cells, which are expressing OE control versus the MFN2. So they increase, they basically transfected these cells with the plasmid, which increases the expression of MFN2. So when this control versus this increase MFN2 expressing CD8 T cells were primed with the so co-culturing with the dendritic T cells, and then these cells were injected in these mouse. So what they found is when there is an increase in this MFN2 expression, so what will happen with the tumor basically. So what they're trying to show is if you increase the MFN2 uh, expression, there will be a less tumor. Here you can see in the PDX model there will be less tumor when these cells were injected inside the PDX patient derived this xenograft containing mouse. Similarly, they have found that in the leaf, for example, here when these cells were injected, these cells directly are going to the PDX compared to the other part of the mice in this case. And they also checked for the tumor apoptosis rate. So the mice which are injected with this OE-MFN2 or the high expressing MFN2 CD8 T cells, there is a more tumor apoptosis. It means that these cells are more efficient to kill the tumor cells in this case. And further, they checked for validated this OE-MFN2 expression. Here you can see here there are more MFN2 expression in these cells compared in these CD8 T cells compared to the control. So this is suggesting now what it is suggesting that um, uh, basically when you increase the MFN2 expression, there is a increased activity of CD8 T cells and there is a decrease in the tumor. If you knock out the MFN2, there is a decrease in the activity of CD8 T cells and there is an increase in the tumor. So this is the line of observation they are following in this paper. Now, increasing MFN2 in CD8 T cells improve the efficacy of cancer immunotherapy. So, for the same purpose, they use this OE MFN2, or we can say MFN2 expressing. Basically, the CD8 T cells were injected when in the CCR. This is the same data which is here. Basically, here they plotted it here. So there is a decrease in the tumor volume. There is an increase in this IFN gamma level and the CD8 T cell percentage, and even they use the increased expression of both MFN1 and MFN2. In both cases, they have found that there is a decrease in the tumor, although the decrease was more prominent in MFN2 expressing compared to MFN1 expressing. Furthermore, what they did is they use this drug, which is an anti-arthritic drug, which is known to increase the MFN2 expression. So you can see here when they're increasing the dose of this drug, there is an increase in the MFN2 expression. And further, they checked for the chelanexin in this mitochondrial ER junction, and they found that there is an increase in the chelanexin in this case, which is suggesting that by any way, if you increase the MFN2 expression, whether it is chemically increased expression, there will be an increase in the mitochondrial junction, which is showing here. Furthermore, when they use this drug in wild type, for example, wild type is here, and the leufelamide was given, for example, so directly in this wild type. There's a significant difference we can see here in tumor growth in wild type. When the anti-PD1 therapy was given, it is here there's a decrease in the tumor growth, but there is a more decrease in this tumor growth when the leufelamide was given with the anti-PD1 therapy, which is suggesting that the increased expression of MFN2 in anti-PD1 therapy will increase the function of CD8 T cells here also. Further, so to conclude this paper, I want to say how these people, how these authors or this research is proceeded. So initially, they tried to correlate this MFN2 expression with the overall survival in the RCC renal cell carcinoma patients. Then they correlated it with the CD8 T cells, that if the MFN2 is more, there is more uh, activated CD8 T cells, and they have shown this in the patient sample as well as in the mice sample. Further, what they did is they wanted to know how whether it is happening, so they correlated it with the mitochondrial function also at the same time. Furthermore, they wanted to know how exactly is happening, whether it is just the mitochondria and ER are coming together, that's why it is happening, or some proteins are involved. Then they found this SERCA2 protein. Now they furthermore go deep in this interaction, like what is the nature of this interaction, and they found that this SERCA2 and MFN2 are basically maintaining the homeostasis in these cells. Furthermore, they checked for the effect of this increase or the decrease in expression of MFN2 on the immunotherapy, and they found that the increasing the expression of MFN2 is good for immunotherapy in this case. So overall, the conclusion, I would like to conclude this paper, and they also concluded this paper because The nature of mitochondrial behavior in tumor infiltrating T cells was poorly understood. So they went with this study and they found that mitofusion 2 expression is positively correlated with the prognosis of multiple cancers. So they checked in the RCC and they also given some references in which they have cited that the same thing is happening with the other cancers also. The genetic ablation of MFN2 in CD8 T cells dampen mitochondrial metabolism and function and promote tumor progression, which they have shown in the mice, in the B16 model, and the other model also. MFN2 enhances mitochondrial endoplasmic reticulum contact by interacting with ER embedded calcium ATPase circuit, which they have shown by using this purified MFN2 tagged with the flag and then their binding partners, and then further the characterize this function by using various variant of this MFN2 and the point mutation they did in MFN2 and expressed it in they thoroughly did this thing. Furthermore, they concluded that the facilitating the mitochondrial calcium influx required for efficient mitochondrial metabolism. So in this the experiment of linker experiment in which they use linker, they have shown that only just increasing this Mito-ER junction or this interaction or tethering is not only enough, because if the SERCA2 is not present there, it will be unidirectional and it will eventually will increase the cell death of the CD8 T cells, because the more calcium will flow inside the mitochondria and the cell will die. So this SERCA2 is required for this optimal function and the flux of this calcium to the mitochondria. However, to stimulate the ER calcium retrieval activity of cell, that's why they are concluded. Elevated mitochondrial ER contact by increasing MFN2 in CD8 T cells improve the efficacy of cancer immunotherapy, which they have shown in the anti-PD-L1 therapy. They use this increased expression of MFN2 and they use this leufonlamide in this case, and they've shown this claim is true. So overall, they concluded that a tethering and buffering mechanism organelles crosstalk that regulates the metabolic fitness of tumor-infiltrating CD8 T cells and highlights the therapeutic potential of enhancing MFN2 expression to optimize T cell function. Thank you for listening patiently. If you have any question, you can comment, and if you have anything to discuss and if you want us to improve something on, you can write it to the comment and we will go through it. There will be more presentations like this presentation, so I already have posted these 3 papers. So this was the first paper which I have posted and which we have gone through as a journal club. The second paper is more about mitophagy, which we will talk about it next Sunday most probably. Thank you for listening.